Hello friends, welcome to my pessimistic guide to anti-aging research, episode 8. Today we will start talking about causes of aging and we will begin with mutations. Just when I was preparing this video, our pastor uploaded his own dealing with the very same topic. Naturally, being lazy by nature, I watched it with some interest, hoping that maybe I will not need to bother at all. Unfortunately, although I wholeheartedly agree that the silver bullet approach to our anti-aging efforts is idiotic by default, I found myself unable to accept the rest of his top-to-bottom vision of the aging process. I'm a bottom dweller, and therefore I will have to go on with my own vision. As I mentioned before, there are more than 300 theories of aging. They are by no means mutually exclusive. They simply describe different facets of this extremely complex phenomenon. From my perspective, everything contributes to aging in interconnected fashion, thus creating a rather messy continuum. Pragmatically, though, there are a few prudent questions that need to be asked before we go any further. First, which of the many contributing mechanisms that we know of are primary and which ones are secondary? For example, cancer, stem cell exhaustion, and senescence are certainly linked to accumulation of mutations and epigenetic changes, and therefore are likely to be derivative. The second question would be which determinant of our lifespan are most proximal, because these are the ones that we need to deal with first. I can envision that multiple processes maintaining our general health may have different expiration dates. Unfortunately, we expire when the earliest expiration date comes up. Depending on individual preferences, it could be mutations, it could be telomeres, it could be growing deficit of neurons, it could be something else. You see, I'm trying to be neutral here. Finally, it would not hurt to know which of the mechanisms are responsible for the biggest leaks of our vitality. Because if they remain in place, plugging small leaks is not likely to produce noticeable results. If we eliminate a small-scale third-rate contributor, what will happen? I will venture to say nothing. 2 small players, 10, still nothing, unless, of course, all-powerful placebo effect fools us into feeling better. One of the good examples that comes to mind is antioxidants. 70 years of fantastic experimental results, public enthusiasm, and $20 billion industry, and okay, I'll try to do some drums. Well, and nothing. It is common to classify things, and I'm going to oblige. Keep in mind, though, that any classification, while convenient, creates rather false interpretation of reality. So, in the broadest sense, there are 3 primary sources of aging. First, accumulation of damage in our blueprint, DNA, which include mutations, epigenetic changes, and probably telomere shortening. Second, accumulation of damage due to built-in imperfections in our systems and processes, even in pristine conditions. None of the biological processes is absolutely perfect, and there are some that may have significant contribution to aging. For example, the recycling of the worn-out components of the cell is taking place in the lysosomes, and it is not 100% efficient. Some large biomolecules have non-degradable parts that tend to accumulate in lysosome and impair their function. Or accumulation of some very detrimental mutations in the mitochondrial DNA can be viewed as a result of an obvious design flaw that Mother Nature neglected to correct. Third, extrinsic causes that remain relevant. For example, our symbiotic relationships with bacteria is a factor that we had not contemplated seriously until recently. With 37 trillion of our own cells and 38 trillion of legitimate resident bacteria, which are found practically everywhere now, it is not even clear what we really are, proud Homo sapiens or glorified carriers. Incidentally, in this case, a distinction between extrinsic and intrinsic becomes very blurred. Okay, now I will start with mutations in the nuclear DNA, where most genes are located, and later in the mitochondrial DNA, which suffers most damage. Because the mutations violate the very foundation of our existence, they have been proposed to contribute to aging from essentially the time when the role of DNA in heredity was established in early 50s. Since then, numerous studies have shown that mutations contribute to aging. For example, genetic defects concerning DNA repair and resulting in accelerated accumulation of mutations are responsible for the appearance of signs of premature aging and early mortality. One of the flashiest examples of diseases caused by aging, or by mutations, is of course cancer. It is estimated that the development of cancer requires from 1 to 10 mutations in so-called driver genes. Although other mutations show less flair for a dramatic entrance, there is no doubt that there is an infinite number of less conspicuous, of course, but functionally crippling combinations of mutations that do accumulate in every single cell of our body. Recently, we acquired the means to detect all somatic mutations in a single cell, with little or no bias using whole genome sequencing approach. It showed that individual human cells can accumulate thousands of mutations. Luckily for us, genes take up only about 1% of the entire genome, which means that only a few mutations hit genes per se. Also, the genetic code and resulting proteins are organized in such a way that makes only about a third of mutations potentially damaging. Nonetheless, while in young organisms all cells are essentially identical and functionally perfect, the accumulation of random mutations with aging creates a mosaic of cells with different obvious or hidden deficiencies. In other words, in aging body, the cells do not function or react to stimuli the same way. Think of a team of sled dogs that gradually start pulling in slightly different directions. Nothing catastrophic, of course, but the entire system becomes increasingly imbalanced. Also, please note that all somatic cells accumulate mutations at comparable rates, including stem cells, neurons, myocytes, etc. On the flip side, judging from the relatively mild level of mutations accumulated in coding sequences, a fair proportion, or maybe even a majority of cells, retain a potential for additional, or maybe even several additional lifespans. That is, if we pretend that other drivers of aging do not interfere at all. Now, let's move from nuclear to mitochondrial DNA. Compared to nuclear DNA, mitochondrial DNA is less protected, and it has many other differences that make it more susceptible to mutations. As a result, the accumulation of mutations in mitochondrial DNA is at least one order of magnitude higher than in the nuclear DNA. Cells harbor hundreds or thousands of mitochondrial DNA copies. Having many copies helps to compensate for mutations in individual mitochondrial DNAs, up to a certain point. Some cells end up having the vast majority of their mitochondrial DNA copies mutated. These cells lose the ability to utilize oxygen and become respiration deficient. They also become functionally impaired and more susceptible to death, which is especially dangerous for slowly on non-renewable cell populations like neurons and myocytes. It has been shown that in elderly humans, up to 30% of surviving neurons and myocytes are respiration deficient due to mutated mitochondrial DNA. This seems to me like a powerful enough and very likely contributor to the development of Alzheimer's and sarcopenia. So, with regard to Alzheimer's and aging-associated dementias in general, maybe instead of putting all eggs into the amyloid basket and with the determination of a woodpecker, insisting on this already failed approach, we should entertain a broader and more biologically sensible perspective. Just saying, don't get too mad. Anyway, in my next episode, I will discuss the epigenetic component of the aging process, which is becoming more and more compelling with every new bit of information. See you then.